In this video, I'm going to discuss developmental changes that happens at different time periods after fertilizations. But in order to understand this table, first I would like to review some basic embryology. So basically, at around day 14 of the menstrual cycle, that's the time that there is an LH surge, as a consequence of which ovulation happens, and what remains of the Graafian follicles is called corpus luteum, which produces progesterone. And then the egg usually gets fertilized in the ampulla of the fallopian tubule. So once fertilization happens, then the zygote will undergo division called cleavage, where there is no growth but rapid division. So one cell zygote will divide into 2 cells, 4 cells, 8 cells, 16 cells, and then 32 cells, which is called morula. And so morula is a mass of 32 cells. Now I should mention that there are some books that refer to these 2-cell stage and 4-cell stage and so on as 2-cell blastula or 4-cell blastula, while there are some other embryology books that refer to blastula as a mass of hollow cells. Regardless what terminology we use, it's the morula that will later on form a blastocyst. So blastocyst is made of multiple layers. The first layer is zona pellucida, which is formed after fertilization, and the function of zona pellucida is to prevent fertilization with more than one sperm. And this zona pellucida will remain in this structure until later stages, while Syncytiotrophoblas starts to invade the uterus. So still at the stage where blastocyst is formed, zona pellucida is present. And then blastocyst contains an outer cell layer that is called trophoblast, and an inner cell mass that is called embryoblast. And so here I have another image that shows you the cavity inside the blastocyst is called blastocele, or the other name for it is blastocyst cavity. And then we have the embryoblast, shown in orange right here, and the outside layer, which is the trophoblast, gives rise to 2 different cells. Cytotrophoblast, which is the inner cell layer, and these cytotrophoblasts will fuse to form Syncytiotrophoblast, which is the outer cell layer, and these Syncytiotrophoblasts are now able to invade the uterus, and they would also produce human chorionic gonadotropin, which is essential for maintenance of corpus luteum, so that corpus luteum can continue to produce progesterone and prevent menses from happening. Now, if there is no fertilization, then there won't be any blastocyst that is formed, and thus, since there is no HCG production, corpus luteum will degenerate into corpus albicans, as a consequence of which there won't be any progesterone production, and then the wall of the uterus starts to shed and cause menses. But then if there is fertilization, then there would be HCG production, which will help maintain the corpus luteum and progesterone production. Now, so far we've discussed what happens to the trophoblast. Next, I would like to discuss what happens to the embryoblast. So let's say here we have the trophoblast. Then the embryoblast will divide into epiblast, which forms the amniotic cavity. And then the other side will give rise to the hypoblast, which forms the yolk sac cavity. So here's another image that shows you how the trophoblast has differentiated into cytotrophoblast, which is the inner cell layer, and then cytotrophoblast that will fuse will form the Syncytiotrophoblasts that are now able to invade the uterus, and they could also produce human chorionic gonadotropin. And Syncytiotrophoblasts will start their invasion on around 6 to 7 day post-fertilization. So it's about 6 to 7 days post-fertilization where there would be start of the HCG production. And then the other cell type, which was the embryoblast, gives rise to the epiblast. And epiblast will form the amniotic cavity, and then the hypoblast, which forms the yolk sac. And then in addition, epiblast gives rise to the extraembryonic mesoderm, which has 2 layers. The somatic layer as well as the visceral layer. So the somatic layer is shown right here. You can see that it lines the Cytotrophoblast as well as the amniotic cavity, while the visceral layer is lining the yolk sac. And so once again, epiblast gives rise to the amniotic cavity as well as the extraembryonic mesoderm. And so extraembryonic mesoderm has 2 layers, the somatic layer, which lines the Cytotrophoblast as well as the amniotic cavity. And then there is also a visceral layer which lines the yolk sac. And then we have the other cells which were called hypoblasts, and these hypoblasts are the ones that gives rise to the yolk sac. And so these 2 layers, the epiblast and hypoblast, are referred to as bilaminar disc, and these are formed at around 2 weeks after fertilization. So if you recall, at 1 week, Syncytiotrophoblast started to produce HCG and invade the uterus, while at around 2 weeks of gestation, the bilaminar disc is formed. Now at the point where the epiblast and hypoblast meet, this is referred to as prochordal plate. So here we have the prochordal plate, and this is the site of the future mouth. Now about 1 week later, at around 3 weeks post-fertilization, the bilaminar disc will start to differentiate into 3 layers, and this differentiation process is called gastrulation. And there are 3 layers called ectoderm, mesoderm, and then endoderm. And then in a separate video, I have just discussed how trilaminar layer will differentiate into other tissues as well as the anterior and posterior neural folds. So I would like to stop discussion of the embryology at this stage and now refer back to the table that we have just started at the beginning. So once again, at around 1 week of gestation, that's the time that the Syncytiotrophoblast will start producing the human chorionic gonadotropin, which helps maintain the corpus luteum. And so corpus luteum will now produce progesterone, and progesterone is required to maintain the wall of the uterus, and so pregnancy can continue to go on. Now I would like to mention that HCG is actually the hormone that is used for detecting pregnancy, and so HCG can be detected in the blood about 1 week after fertilization. And then in the home urine pregnancy test, it can be detected about 2 weeks after fertilization. So in about 1 week after fertilization, you can detect human chorionic gonadotropin in the blood, while about 2 weeks later, you can detect it in the urine, which is used as the basis of the home urine pregnancy test. Then we just discussed at around 2 weeks of gestation, a bilaminar disc will be formed, which is made of the epiblast and hypoblast. And the way you can memorize it is that by week 2, a bilaminar disc is formed. And then at 3 weeks post-fertilization, gastrulation happens, and so a trilaminar disc will be formed, which is made of the 3 germ cell layers, the ectoderm, mesoderm, and endoderm. And again, it's easy to memorize that. Trilaminar disc is formed at 3 weeks post-fertilization. Next, we have weeks 3 to 8, during which period there is organogenesis, where multiple different organs will start to develop. And therefore, during this stage, the embryo is highly susceptible to teratogens. So if they provide you a list of different time periods and ask you during which period embryo is most susceptible to teratogens, which will cause defects in the development of organs, the answer would be between weeks 3 to 8 post-fertilization. Now at around week 4, the heart will start to beat, and also the limb buds will start to form. And the way I memorize it is that heart has 4 chambers, and we also have 4 limbs. We have 2 arms and 2 legs. So at around week 4 post-fertilization, the heart, which has 4 chambers, start to beat, and we develop the 4 limb buds. And then finally, at around week 8 of fertilization, fetus will start to move. So once again, let's review week 3 to 8 one more time. So we have organogenesis at around 3 to 8 weeks of gestation. At week 4, we have the heart to start to beat, and then we have the 4 limbs buds that are being formed, and then at week 8 of gestation, fetus will start to move. And then we have week 10, during which period the genitalia will start to develop male versus female characteristic. And finally, around weeks 20 to 30 post-fertilization, that's the time that brain gyri will be formed. So if you look at embryo, at around 4 weeks, there is a very primitive central nervous system that is there. So here we have the optic vesicle, which will develop into the eye. We have the forebrain, we have the midbrain, and then we have the hindbrain. At around 8 weeks, it's more developed, so we have the forebrain, we have the midbrain, and then hindbrain, and then the spinal cord, and then finally it's at around 20 to 30 weeks gestation when the brain gyri will start to develop. Alright, and that concludes our discussion of different developmental changes that happens during different time periods post-fertilization.